Hello everyone, welcome to Microbial Chronicles. Today we are going to delve into the microscopic world. Now, if Sherlock Holmes was a detective, then he would identify the criminal by means of crime staining. So today, let us learn a little bit more about this procedure and what it can be used for and how we can identify the various bacteria based on this. Now, this was a method which was originally developed by scientists called as Hans Christian Gram, therefore gram staining. Now he discovered this accidentally. So the bacteria can be divided into two types, gram positive bacteria and gram negative bacteria. Now the main difference in these two types of bacteria is their cell wall. So before we move further into understanding gram staining, let us first have a look at the cell wall. So what are the main differences between the gram positive cell wall and a gram negative cell wall? The first and most important is that the gram positive cell wall has a thicker peptidoglycan layer. Now, if you remember, if you understand the anatomy of the bacteria, cell wall is the outermost layer, right? So now the cell wall has more peptidoglycan layer, therefore a thicker peptidoglycan layer basically, which will help to retain the dye more strongly. That's why the gram positive bacteria they appear purple, and the gram negative bacteria has a less thinner peptidoglycan layer, so they retain the dye less strongly, therefore they are more pink in color. This is a very important difference. Next difference is that the lipid content in gram positive cell wall is nil. It does not have any lipids at all, whereas a lipid content is present in the gram negative cell wall. Now in the gram positive cell wall, the teichoic acid is also present, but in the gram negative cell wall, the teichoic acid is absent. Now in the gram positive cell wall, there are a few amino acids variety compared to the gram negative cell wall, which has a several amino acids variety. So these are a few important and basic differences between the gram positive cell wall and a gram negative cell wall. So moving on, let us look at which all are the materials which you will require. The first and most obvious one is a specimen from which you are going to make a slide, and a clean slide obviously. So we will be making first a smear. Now apart from that, the materials required for the gram staining is a primary stain. A primary stain is mostly we use a crystal violet, but it can also be methyl violet or gentian violet. Now I will explain about the function of each of these stains and the types of bacteria and examples of each and how to remember them easier in the next video. In this video we will focus only on the tutorial of how to perform the gram stain. The next step will be a mordant. A mordant is required. A mordant is like a fixer. So this is the grams iodine. Okay, so this is important. The third item after this which we will require is a decolorizer. Now a decolorizer is the most crucial step. So we can use acetone or we can use ethyl alcohol. The duration you keep each will differ because depend on that, which I will explain more in the procedure. So just to reiterate, the decolorizer is the most crucial step. And lastly, we will require a counter stain. The counter stain basically provides the background against which we will view the slide under 100 times oil immersion lens of the microscope. So the counter stain we use in gram staining is safranin, or we can use a stain which we use in acid fast staining, which is dilute carbonyl fusion. Acid fast staining will be covered in another video. So these are all the materials which are required to perform the gram staining. Now let's actually perform the procedure. So to understand this, so first we will heat fix the smear. So first to make a smear onto a clean glass slide and heat fix it. After that, do the procedure as follows. So the first step is to add the primary stain. So after heat fixing, we have to add the primary stain. So the primary stain, as I already has told, is crystal violet or methyl or gentle violet. In our laboratory, we use crystal violet, but any of the above is fine. So how long do we keep this? We keep this primary stain for a duration of 1 minute. So this has to be timed exactly. So better to time it on a watch or your phone on the timer, but it has to be exactly for 1 minute. The timing is really crucial in this gram staining or in any staining procedure basically. After that, what has to be done is we will wash with water or rinse with water. Then we will add the mordant, which is grams iodine in this case, and this we will keep for a duration of 1 minute also. Again, this also has to be timed. After that, again we will rinse with water. Then we will add the decolorizer. Now decolorizer, I will again stress, is the most crucial step. Now there's a reason why the timing has to be exact over here, because if the timing is exceeded, that could decolorize your bacteria. So let's say the gram positive bacteria, but if it's decolorized too much, they will appear gram negative, and this can lead to errors in diagnosis tomorrow, and that is really not acceptable. Therefore this duration has to be exact. For acetone, 2 to 3 seconds, and with 75% ethyl alcohol, 20 to 30 seconds. So again, I'll reiterate once more, the timing is really crucial. After that, we will rinse with water, and lastly we will add a counter stain, which is safranin, for about 30 seconds. Then again we will water rinse, and then finally we can dry the slide. Either we will air dry it or we will pass it over a flame, okay? But the only thing to make sure is that we don't heat the slide too much so that it cracks. So once it is dry, it is now ready to be focused under a 100x lens of a microscope. So I hope you all have understood the procedure, and with this we will end today's video. I will be explaining the remaining part about gram staining, as in the principles of gram staining and the function of each of the components in this procedure, and how to remember the names of gram positive and gram negative bacteria with some mnemonics. So all this will be covered in the next video. So this is my first video, and I hope I was able to explain it well. I would really appreciate any feedback, so you can please like and subscribe, and yeah, I'll see you all in the next video. Goodbye and have a nice day.